Hi dear students, welcome you all on the behalf of Rahul Anand Biology tutorials. So, as you know that I am Rahul Anand and this is somewhat around my channel whose name is actually the Rahul Anand Biology tutorials. So now, let's begin with the very interesting topics which are often being asked in terms of your Neet, often the Aiims, often the All India Pre-Medical Examination. So these are literally belonging from which of the chapters of your book? So these are belonging from the biomolecules. So today here we are going to study about what are actually the enzymes. So enzymes are what? So enzymes are the biocatalyst which increases or decreases the rate of reaction without being altered. Now, so let's understand about what are actually the binomial and classification of your enzymes. So these are oftenly being divided in terms of the three broad categories. So these are what? So these are the lyses, then the here. So particularly the lyses, then what? Then the isomerase, then what? Then the ligase. So literally, what are actually these things means in terms of your lyse? So in the case of the lyses, what will be happening? So in the case of the lyses, the removal of group will be occurring without what? Will be occurring without hard lysis. Often when we are talking about the case of the lysis, so in this case, what are the other respective? So in the case of the lysis, the breaking of the covalent bond literally occur. Often when we are talking about the case of the lysis, so in this case the here merit of the cleavage of the covalent bond will be literally taking place or either occurring. Find here. So your lysis literally catalyzes the cleavage of the covalent bond. So catalysis means what? It's these are nothing but the particular case of the here meant of the biocatalyst or either the bio agents which are playing important role to increase or decrease the rate of reaction without particularly being altered. After that, we are often having the certain specific role, such as what? Such as when we are talking about the case of the lysis. So your lysis literally react to the C single bond C, then the C single bond O, then the C single bond S, then the C single bond N, or either often with the another reaction, such as what? Such as the C that, and after that, when we are talking about the particular examples in it, so in this case we are having the histidine decarboxylase. So when your histidine decarboxylase are literally being treated in the presence of the C single bond C in the histidine, it results in the formation of the carbon dioxide and the histamine, often the aldose. So particularly when we are talking about the specific reactions, so in this case we are having what? So in this case we are having the fructose 1,6 what? So in this case we have the fructose 1,6 phosphate. After that, what will be happening? So when these are particularly being treated in the presence of the aldose, it results in the formation of the dihydroxyacetone phosphate plus glyceraldehyde phosphate. Often the subdivision of the aldose are of course being seen because these are what? So these are the catalysts or either playing important role just like the enzymes. Now, so after that we are often having the another specific resemblance of the isomerase. So your isomerase literally do what? So isomerase are what? So when we are talking about the isomerase, so it catalyzes what? So it catalyzes the isomerase reaction, or either you could say that in the case of the isomerase, the particular reaction of the isomerization reaction will be taking place. After that, it literally catalyzes what? So it literally catalyzes the cleavage of the arrangement of the molecular structure in order to form what? In order to form your isomers. So after that, literally what will be happening? So after that, the literally process of the isomers will be literally obtained. Such as in this case we are having the example of what? Of the phosphohexoisomerase. Of course, then what? So the phosphohexoisomerase, then what? Then the here meant of the maltase. Find here the phosphohexoisomerase, the maltase. Often the another symptoms or either the examples which are literally being enrolled in it. So these are what? So these are the epimerase. Of course. So we are often having the case of the epimerase. Thus when we are talking about the particular reaction, so in this case what will be happening? So in this case the glucose 5-phosphate will be treated in the presence of the Isomerses in order to form the ribulose 5-phosphate. After that, your here meant of the lysis process literally takes place. So in the case of the lysis, the formation of the bond will be open with reference to what? With reference to the ATP cleavage often will be occurring or rather takes place. Thus we could say that in this case of the particular reaction, what will be happening? So in this case of the particular reaction, when the pyruvic acid are literally being treated with reference to the carbon dioxide, so it results in the formation of the oxaloacetic acid. What I have told? So I have told that in this case, when the pyruvic acid are literally being treated with reference to the carbon dioxide, so it results in the formation of oxaloacetic acid in the presence of what? In the presence of your enzymes. Of course, so this is the particular process when we are talking about the case of the here momentum of what? Momentum of the ligase. So the nomenclature and classification of enzymes are being divided into the three to four broad categories in which we have dealt about what are actually the lyses. Then what? Then the here momentum of the epimerase. Then what? Then the another reaction such as the isomerase. Often what? Often the another fact such as what? Such as the ligase. So this is somewhat the condition when we are talking about means what are actually the enzymes. So enzymes are what? So these are actually nothing but the biocatalyst which increases or decreases the rate of reaction without particularly being altered. So if you like the work with reference to the education as well as the lectures as well as the videos, so you could often subscribe, then particularly like, then comment below in the text. Thus I'll be knowing that what concept actually you are hungry for or what event you understood or rather got. So very thank you with reference to the Rahul Anand biology tutorials.